So I guess I'm going to talk about how compromises shape the design of biological systems. So I was invited, oh, there we go. I was invited to give this talk, and my first thought is, how am I going to make sure I'm not too boring? Because the problem is, most of you are here for AI. This is the Sussex AI seminar. I do some AI adjacent stuff, but primarily my interest is in science, neuroscience. So actually, it seems like quite a few of you are neuroscientists. So there will be some intersection with people who are interested in what I do potentially, but it's not going to be complete. Furthermore, the number of people who are going to remember all the research details in a week is going to be that much smaller. So I thought, what can I usefully offer with you? I will offer some science, don't worry. I'm not just going to talk about random stuff for an hour, but I thought I'd give a couple of thoughts first on career transitions for the younger researchers in the audience. Some thoughts on doing science as somebody from a non-scientific background, from more of an engineering maths background, which I think is something that resonates with people coming from AI. And of course, at least some research, but I'm going to try and emphasize style over substance and stick to stuff that's not too neuroscience-y. It will end up being a bit neuroscience-y. I make no apologies for that. You saw the abstracts. So the first thing I'm going to quickly just go through, for the benefit of people starting research careers, this might be a bit boring for older members of the audience, is how did I end up in neuroscience? And the reason is not because, as I said, I'm a particularly interesting person, but I would like to give you an extra data point about what career transitions look like and why meandering a bit, not too much, can be stressful in the moment, but useful in hindsight. So I started by doing an undergrad in maths, and I kind of liked it. And I thought I wanted to go into research and do a PhD. So I asked my undergraduate supervisor, who I did a project on some pure maths thing with, like, hey, I'd like to do a PhD. And he kind of basically said, you know, my field doesn't actually have much going on these days. So I was like, okay, fine. What about machine learning? So I asked my pastoral tutor, hey, I'm interested in doing a PhD in machine learning. And he said, this is a very muted version of what he said, but something like, don't do a machine learning PhD. They're stupid and you'll never be a proper mathematician. So I thought, okay, I won't do a machine learning PhD. Thought about computational neuroscience. So I applied to someone whose research seemed interesting. And he said something like, no, don't bother applying to me. Getting a first is not enough from a university rank like yours if you haven't got any extra stuff. So I was like, okay. The point is not that these are all very intelligent people. They're all prominent academics, follows the Royal Societies, and they have good opinions. But excessive advice can be stressful, and you always have to consider the background and biases of the person talking to you. Because in hindsight, any of these options would have been quite nice, I think. So I ended up going to systems biology, which is kind of like, well, I'm going to talk about it in a second, but it was a doctoral training center. So that's a system where you do 1 year of taught courses, and then you do a 3-year PhD. So what is systems biology? I was kind of interested in biology in general. I'd done some mathematical biology courses. Systems biology is a feeling, in the sense that from the 70s to the 90s, from the 60s to the 90s, last half of the 20th century was called the golden age of molecular biology. There was all this brilliant experimentation where we've really figured out the components of the cell, what they are and what they do. So DNA, the structure of DNA was one of the earliest ones. Then the ribosome, which is, you know, like a 3D printer that prints proteins from DNA, all sorts of other stuff. And systems biology is a little bit more about how do these things work together? Because in the end, the cell is a really complicated system with loads of components, loads of proteins, genes, metabolites, all working together in harmony, or sometimes not in harmony. And at the time I did my PhD, there was a little bit of a hype in systems biology, a little bit like the AI revolution today. Maybe not, well, obviously not as much hype as the AI revolution today. But there were some parallels because there were two separate advances that were justifying this hype. So one was an increase in the ability to collect data. This is called the omics revolution. So in the course of your studies or Wikipedia, you might come across words like proteomics, genomics, metabolomics, transcriptomics. What this means is simultaneously measuring many of the things you're talking about at the same time. So proteomics is being able to measure in a cell many different protein concentrations at the same time. Similarly, genomics. And there were just loads of technologies coming out to be able to get large amounts of data in a cell. Simultaneously, computers were getting better. And we were thinking, hey, we can actually try and model these things and build big computational models of overarching systems within the cell and in tissue. And that was exciting. For instance, this was the paper I did we did our first journal club on in the DTC, and this kind of captures the zeitgeist, the whole cell computational model. So this is the picture of the model. Each component in this is itself a massive model. Like the whole model had thousands of states, and it was an active topic of discussion. Like, are these things meaningful? Are they not meaningful? What can we get from them? Now, there are some problems when you're building big models. So one thing is when you get a really as the complexity of your model you're building increases, its ability to fit data also increases really fast. And this is captured nicely in von Neumann's quote, which is with 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk. So that's really great for AI. Like you have massive models with loads of parameters, and that's because AI is used for prediction. Your AI system is trying to fit any data really well. It can make it hard. I'm not saying it's an insurmountable challenge, but it can make it hard for interpretation. It's hard to actually interpret what's going on inside a big neural network. Similarly, if we look at these massive models of metabolic systems and proteins and things in the cell, it's sometimes hard to figure out, are they correct? Are they wrong? What does it mean to be correct? If you kind of know a priori that because they're so complicated, they're going to be able to fit your data, whatever happens. Another thing I found difficult was the noisiness of data in biology. And again, this is not a criticism. This was just me coming from maths. You know, like biology things look kind of like this when you look at papers sometimes. And when you can make a constellation out of your data points, that's, it's easy to fit it usually. This does not at all void systems biology. It's a really interesting field with lots of stuff going on. But I was kind of young and coming from maths, and I'd kind of gone really I was really into oh, wow, I'm going to model everything. It's going to be great. And then I saw these kinds of problems, and I thought, oh, no, this is all terrible. I'm just going to leave systems biology within about 2 or 3 months. I regret that, actually. I've got a more nuanced view of this. There are good papers, and there are bad papers, obviously, right? But I learned a couple of lessons that I think are important. So one thing is, good science can require complex models. It often does. But building complex models is not equal to good science. And I see this a lot. Sometimes when you're coming from a maths background or an engineering background, you're like, let's model everything, let's build it. You really got to think about why you're doing it, I think. Second, fashionable papers don't usually define fields. So, I'd see a couple of, Nature papers and I'd think wow, this looks amazing. Then I'd look at the methods and I think, oh, what's going on? Well, this must mean the whole field is terrible. But actually, no. You get lots of fashionable papers with some good bits, some bad bits. That doesn't define a field. And finally, watch out for oversteering. If you really like something and then you really dislike something, the truth is in between. So then my objective was to keep my PhD funding, which was for systems biology, but run as far away from biology as possible. So I did something, I went to a field called control theory, which was the optimal way of doing that at the time. Control theory is the science of designing systems with feedback, allowing them to regulate themselves. So for instance, the easiest example is cruise control in the plane. The plane gets loads of wind gusts and stuff, but it kind of autocorrects, so the pilot doesn't have to do all these things. That's control theory. Now, 1 or 2 things about control theory. It's a really well-developed field in the sense that the sophistication of the mathematical frameworks is really high. To be able to do theory and control, you need to just learn a lot of maths. And do lots of proofs. On the conversely, the theory is ahead of practice, which is very unlike AI. So in AI, we often have these stunning practical results, and then theorists are running to catch up and explain oh, we expected this all along. Whereas in control, a lot of the time, you're building theories and principles for things that are never actually potentially going to be realized, or at least there's lots of practical hurdles to building them. This was bad for me because I've done an undergraduate in maths, and you learn in undergraduate that hard maths means success, and you want to do hard maths, and that's great. And then you kind of implicitly take that bias to postgrad, and you think hard maths means hard research and interesting, useful research. But actually, again, control theory has good and bad papers like anything, but it's very easy in a field like that to accidentally go into a kind of theoretical hole where you think you take a step out and think, oh God, what am I doing? So I struggled hard to write my difficult maths paper, and then I published it, and I was happy, and then I took a step back, and I thought, oh God, what was the point of this? So I had an existential crisis and completely changed my topic in the third year of my PhD. So don't make that mistake as well. Finally, I went into a postdoc in computational neuroscience. Why? It was really an accident. I just went to a conference in control theory, and sitting next to me was a neuroscientist who wanted to do control theory, and he was trying to talk to them. And we just had a nice conversation over dinner, and I was thinking a bit about what postdoc I wanted to do. And I thought, wait, why don't I just try neuroscience? And he became my postdoc supervisor, and that was great. And actually, I learned more control in many ways doing neuroscience than I did doing control. Because in control, I had to write all the proofs, but I didn't have to explain simple concepts intelligently. And I couldn't actually at that point. I still can't do it completely for control, but I got better at that kind of stuff during the neuroscience postdoc. Cool, that's enough talking about me. That was too long probably, but fine. So now I'm going to go to some scientific questions. I'm going to try and, as I said, emphasize style over substance and give you a flavor of the type of research I like to do. So the first question I'm going to ask is, when does a synapse stabilize? So some of you may not know what synapses are. I think most of you probably do, looking at the audience, but fine. So think about an artificial neural network. We have these units, like a ReLU unit or a sigmoidal unit, and they're connected to other units. And those connections have a strength, which we call the weight. Training is changing the weight. So when you're learning training on the data, you're basically changing the weights until you finish learning, and then you stop. So you could think of the weights as holding a memory of all the stuff you've learned. I could download ChatGPT's weights, and then that's all the information needed to use ChatGPT. It's got a memory of everything it's learned. The biological nervous system is not that much like an artificial neural network in the sense that the neurons are a lot more complicated, they're doing a lot more stuff, and the connections are also a bit different. But at the very abstract level, there are strong similarities. You have neurons, they're connected by what are called synapses, so these are the weights. Those synapses change their functional properties, for instance, the coupling strength between neurons, and that's kind of analogous to changing the weight in an artificial network. And so when we change the weights, we call it synaptic plasticity. And most of our theories in neuroscience think it's synaptic plasticity that is responsible largely for learning. So you're learning something, your synapses change and store the learned information, and you're done. Because that kind of implies that memory is a configuration of synaptic weights. And to have good retention of memory, you want to have stable synapses. If I take an artificial neural network that's trained and I just randomly change some weights, I'm going to destroy the information. I'm going to destroy the memory. That would presumably be similar in a biological nervous system. That would be really interesting from a scientific point of view, because maybe as a scientist, then I can actually image neural circuits in vivo in a learning animal and say, oh, look, in this bit, the synapses aren't changing much, so it's probably holding a memory and not learning stuff. Here it is, the synapses are changing, so maybe it's in a phase of learning. And that would help us understand what's going on and what's learning which bits in the brain. And the imaging modalities are actually getting to the point where we will be able to do that. We already can in some animals like fruit flies, kind of. So I'm going to ask a question. What makes synapses change? So I'm a synapse. I'm connected to a neuron before me, the presynaptic neuron, and a neuron after me, the postsynaptic neuron. First of all, a synapse is a tiny bag of jelly and proteins. So one estimate for cortical neurons in a rat is every day, about 4% of the weight of the synapse, of the protein content, is degraded and replenished. Things are constantly breaking down, and things are constantly being shuttled back, like new components, new building materials, if you like. And that's a process that takes ages and is quite hard to do, because you can't produce the components of a synapse in the synapse. You have to produce it somewhere else, largely. And when you're at that tiny length spatial scale, molecular noise is a massive factor. So it's hard to keep something stable when things are small, because when you look at things through a really, really big, really powerful microscope, things are just fluctuating all the time, from Brownian motion, thermodynamics. So there's all sorts of just regulation processes going on and noise processes just to keep a synapse stable, if indeed a synapse is being kept stable. Meanwhile, synapses are also changing in structured ways due to learning, right? We said that synaptic changes, synaptic plasticity is the substrate of learning. We usually think of that as coming from information from the presynaptic neuron and the postsynaptic neuron, electrical activity. So they spike, and that information is relayed to the synapse, and the synapse says, oh, I should do something about that, I should change. So it would be interesting to think, how big are these two terms relative to each other? Is a synaptic change dominated by intrinsic processes like molecular noise or little homeostatic regulation mechanisms within the synapse and decoupled from the network? Or is it maybe governed by things coupled to the network? This is a question you cannot unambiguously answer experimentally, but there's a couple of ways people have tried to answer this question. I'm just going to show you one of them. So unlike artificial neural networks, if you take two neurons that are connected, they're usually connected more than once. They often have 1, 2, 5, 20 synaptic connections. Sometimes those synaptic connections are far away, but sometimes they're right next to each other. And these are called commonly innervated synapses. So both synapses have the same presynaptic and the same postsynaptic neuron. Now, if we look at their changes over time, and the changes are almost the same, that's imperfect, but a reasonable way of saying that, okay, most of whatever processes are causing them to change are coming from information from the network, because they're probably getting quite similar information from the network. They have the same pre and postsynaptic neuron. They're spatially located at the same place, so they're just getting the same stuff, same neuromodulators, same hormones. So that implies there might be a common network-derived cause for the changes of the synapses. That's not the case in this paper, for instance. So they looked at in vivo and in vitro cortical neurons. I won't go into the details, but basically each point on this graph represents a pair of co-innervated synapses. And they're measuring approximately through their fluorescence the changes in the sizes and the strengths of these synapses. There's a correlation, but it's not a great correlation. So that means that there's quite a large component of changes in one of these pairs of synapses that are not shared with the other one. Statistically, they went from there to claiming that a majority of synaptic change, so 56 plus 8% or something, is unexplained by the patterns of neural activity at the pre and postsynaptic neuron. It's presumably due to intrinsic synaptic processes. That might not be, it's not a perfect experiment, but at least we cannot correlate this change with anything. It's just the synapses just doing their own thing. Another way of looking at it is, again, an imperfect way of doing experiments, but you can also use particular chemical or electrical or optical ways of suppressing neural activity. If you suppress neural activity, maybe there should be no plasticity, because neurons that fire together wire together. Plasticity is usually thought of as coming from patterns of neural activity at the pre and postsynaptic neurons. But actually, in a lot of different preparations, in a lot of different experiments, they've tried this and they just can't get the synapses to stop changing. Usually they can reduce it by a bit less than half. So across systems across systems and in different experimental modalities, it seems that just over half of synaptic plasticity seems to not be coming from network-induced causes, but just being intrinsic to the synapse. Maybe it's noise, maybe it's something systematic, but it's not related to other stuff. Now, why is this surprising? So Donald Hebb's famous maxim is that neurons that fire together wire together. And it's a simplification, but it accurately captures some aspects of how we think learning occurs. So we have some learning experience, like I'm practicing the piano. That sensory experience translates into patterns of neural activity in my brain, different neurons spike. That pattern of spiking is transmuted through some learning rule, Hebbian plasticity, STDP, whatever you want. If you don't know those words, don't worry, into some type of synaptic change. So the synapses are listening to the patterns of activity at their neighboring neurons and changing based on that. But that should mean that plasticity should not disrupt previously stored information, because again, as we said, like the memory should be stored in the weights. So any further plasticity, once I've learned how to play the piano, any further plasticity in those brain regions should be done in a way that doesn't disrupt stored information. So it has to be coordinated in some way across the network. If I just take a random weight and be like, oh, I'll change you, and then that one and say, oh, I'll change you, that will degrade information. I'd have to have a coordinated mechanism to preserve information. I had to say, oh, I'm going to change you, but then I'm going to change you to compensate. So this seems a bit weird, because plasticity doesn't seem to be coordinated across the network. Most plasticity just seems to be intrinsic, autonomous to a synapse. So overall, the problem we have, it seems across preparations, is that what I'm going to call synaptic fluctuations, these intrinsic processes inside the synapse seem to be contributing a larger share of plasticity of changes Then what I'll call systematic plasticity, which might be a learning rule, but more generally is just plasticity that's coordinated across the network by neural activity or otherwise. So as a theorist, I want to ask why? Can I get a good explanation for this? But here's where I hit some of the problems intrinsic to computational neuroscience. I don't know what's being learned. Like I'm just looking at either some neurons in culture or in a particular, or in vivo sometimes, but I don't know that this brain region is learning exactly this. I don't really know the circuit structure. We don't know which neurons are connected to which other ones. We're looking at mammalian cortex, which is just very complicated. We don't know the learning algorithm, like what pattern, how exactly is neural activity translated into synaptic changes? More generally, computational neuroscience is a very nascent subject, which makes it exciting, but makes it frustrating at times. And I think that's well captured with C. elegans. So C. elegans is this tiny microscopic worm, nematode, not really a worm. It just kind of crawls around doing its thing. And it's the simplest nervous system that we study commonly in neuroscience. It only has 302 neurons, and they all have names. We have the full connectome, so we know which of those 302 neurons are connected to which ones. We even have some idea of how strong those connections are. We have a really good ability to characterize the behavior and the neural activity at the same time. So we can see what's going on in the brain as the worm is wiggling around or finding food, all that. So we can do large scale recordings in these freely moving animals, but we still don't have a model of satisfying kind of computational model of C. elegans. We don't have an algorithmic description of how the behavior arises from the neural circuits. And there's all sorts of reasons it's really hard, because you think 302 neurons, that's not that bad, I could do that. But then you think, okay, well, this is being modulated. There's all these chemicals that are modulating their activity. There's probably about 150 neuropeptides, they're called, that change their behavior. If you try and incorporate those, then, well, those are produced by the metabolic system in the cells. So suddenly you're coupled to the whole metabolic system, then you're coupled to the genetic system. Meanwhile, it's an embodied creature in quite a mechanically complex environment. All these things just make it really, really hard, even for a really simple nervous system like C. elegans. Cool thing about C. elegans is the neurons don't even spike, they're analog. So how do we answer scientific questions in a nascent field? I liken our situation to that of a caveman trying to understand a Ferrari. Ideally, we want to understand what the system components are, so the drivetrain, the gearbox, all these things, and how they interact to produce functionality. And this is a worthwhile endeavor, and we're doing this slowly in the brain, but that intimidates me too much personally. So I try and go for a slightly more modest question usually, which is instead of actually trying to figure out how it's built, understanding why it's hard to build. What are the competing objectives and constraints that guide design? So I've spent a lot of time with engineers in my past. And you know what? A Ferrari looks quite similar to a Tesla on the surface, sports car, even though they're pretty different under the hood. And that reflects that there's the same kind of engineering compromises and design trade-offs they have to do because they have similar functional purposes. The same thing happens in nature. That's the principle of convergent evolution. A dolphin and a shark have lots of functional and high-level similarities, even though, evolutionarily they're so distinct. For instance, suppose this caveman doesn't know how the Ferrari works, but he can compare it with a lorry. You can immediately notice that, the Ferrari is designed for something. It's sacrificing a lot of storage space, but it's got really good aerodynamics. Whereas a lorry is the opposite. It has loads of space and terrible aerodynamics. And that gives you some idea into the different functions of those things and maybe explains how the different functions reflect in the morphologies. And if you have that, then maybe you can actually make some predictions about what a new vehicle the caveman sees is actually trying to do. So what I like to do is to try and relate design trade-offs to function without trying to be too specific about the actual components and algorithms going on under the hood. So I'm going to try and show this with two old pieces of work. I apologize, they're quite old, but I thought they, first of all, they're a bit less neuroscientific. And second of all, I think they show this approach easily. And third of all, I have well-trained slides in them. Okay. So let's recap where we were before we went off on a tangent. So I'm a synapse. Most of my changes seem to be happening due to intrinsic processes like noise, things that are not coupled with the network. And I want to explain why this synapse is like that. I don't know what's being learnt. I don't know the circuit structure. I don't know the learning algorithm. So instead, I'm going to ask what functional goals for a neural circuit and design constraints should imply this? Why should I say, oh, of course it's like that, regardless of how it's learning to some extent? So I'm going to think about a very abstract neural circuit. It could be artificial, it could be biological, acquiring and storing experience. So I've learned something, and the memory is stored in the synaptic weights of this circuit. Now, there's these intrinsic synaptic processes called synaptic fluctuations, which are going to degrade information storage, as I explained previously. And I'm going to assume there must be some active compensation mechanism, a systematic plasticity term that's counteracting this degradation. It's cleaning up the mess. You could call it a learning rule if you like. And it's improving information storage. So these two things balance out. And there's some steady state level of information storage. If you like an equation, we can think of W as the vector of all the weights of the network or whatever other adaptive things there are. And it's changing in time. So it has a time derivative, d by dt, which I denote with a dot. And I'm saying it has two components. It has a systematic plasticity term, so something coordinated across the network to keep the information preserved well. And a synaptic fluctuations term, which, as I said, is just a kind of noise term, if you like, something intrinsic to the synapse that degrades information storage. Now, I'm going to introduce a more abstract concept. So from AI, you may be familiar with the concept of a loss function. This is the same thing. I just call it a task error function for no good reason. So if a system is trying to learn something, we can quantify how well it's learned something with some notion of task error. I don't know what notion of task error because I don't know the task. I don't know if the system has access to this task error. But in a sense, learning is improving some notion of error. So implicitly, there is a notion of error. So I can draw this as a graph. So the z-axis is the amount of task error. So if it's high, it's bad. And if it's low, it's good. And the x and y axes are the adaptive elements in the system. So synaptic weights, if you like. So there are going to be some synaptic weights. Can you see my mouse? Yeah, you can. Okay. There are going to be some synaptic weights which correspond to good information storage, and that's corresponding to being at a low point of this error landscape. Bad is going to be in a high point. Plasticity is going to move us along this landscape, and hopefully we're going to be in a good, it's going to move us to low bits of the landscape. That's learning, and I'll go through that in a sec. So the nice bit about this is that it's kind of abstract and implicit. I haven't said anything about the actual circuit or what's being learnt. I'm just saying that there's some abstract mapping from synaptic weights or even other things that are adaptive to some notion of task error. The problem is I have no idea what the landscape is, right? I don't know what the task error function is. I can't just look at a bit of brain and say, oh, this is doing its job well, or this is what it's trying to learn. I'm just going to give a bit more insight here. So if we think of learning, as I said, is kind of decreasing some notion of task error over time. That's the definition of learning, really, right? So that corresponds to moving gradually along the landscape in a direction that decreases it. So this is a bird's eye view of the landscape. So it's kind of like those geographical maps. Learning might not go straight to the bottom, but it has to eventually, with its twists and turns, get to a lower point in the landscape. That's just the definition of learning. On the other hand, what we've been talking about is not learning. We're talking about retaining previously stored information. So I've already learned something, and I'm trying to keep it as best as possible. So on the landscape, that might correspond to just staying in the same place. That would be no synaptic plasticity. I've just frozen the circuit. Or it might correspond to moving around, but in average in a direction that doesn't increase or decrease task error much, because there's this degrading mechanism and this compensating mechanism, and they balance out at some level. So now what I'm going to ask is, let's say we have some unavoidable quantity of synaptic fluctuations, of intrinsic synaptic movements that's going to degrade information storage. How much systematic plasticity should I apply to optimally compensate for it? Should I have loads? Should I have a tiny bit? And I'm going to start by just doing a really simple simulation in the least biological network you can imagine. It's just a multi-layer perceptron, so the simplest artificial neural network. What I'm going to do is before time 0, I'm going to completely train it. So it has 0 error. It's perfectly doing the task it sets. Okay? And now When I start the simulation, I'm going to change the synaptic weights due to two things. One is going to be just Gaussian noise. Okay? So obviously alone, that's going to corrupt the task error. If I have a trained network and I just change all the weights by consistent Gaussian noise, it's going to corrupt the memory. Simultaneously, I'm going to give it a learning rule, which is just going to be backpropagation because that works. So at each time step, I'm getting some learning and some unlearning. And what I'm going to vary is the relative quantities of these things. So I'm going to keep the level of unlearning, the synaptic fluctuations, the noise steady. And I'm going to gradually step up the amount of learning, the systematic plasticity. Where do I get the best task error? Where do I get the best preservation of the memory? Counterintuitively, it's where there's less systematic plasticity than synaptic fluctuations. So it's like the learning-induced plasticity is less than the amount of noise. Bit weird, potentially. You might think that you'd want to have loads of systematic plasticity to drown out the noise, but actually too much is bad. So yeah, best retention when synaptic fluctuations outcompete the systematic plasticity. Now, that was a really silly artificial neural network that doesn't bear any relation to real life. So what I want to do is turn that into a more general phenomenon theoretically and say, hey, this is going to happen even in a complicated biological circuit where I don't really know what's going on and I don't know whether it's white noise and I don't know what the learning rule is. So I'm not going to go through the maths of that in detail, but what I'm going to do is try and give the intuition of how the mathematical argument I made for that runs. So this is a slice of the loss landscape again. So we're at a point in the loss landscape. And I remember that going lower is like learning, it's getting better at retaining the memory. Going higher is getting worse, it's increasing the loss. My overall movement is a combination of the systematic plasticity, which is going to be going downhill in general because it's learning, it's helping, and the synaptic fluctuations, which could be going anywhere at all because they're just random. Or might not be random, but random with respect to the loss landscape. And the net plasticity is going to be the sum total of these two contributions. Now I'm going to make one point that is hopefully intuitive without any maths, which is that in general, if you're in a valley, going in a highly downhill direction, there's usually more upward curvature than a random direction. Makes sense? Like if I'm looking at the bottom of a valley, it's kind of a U shape, whereas if I'm taking a cross section in a different direction, it's less of a U shape. So I kind of formalized this a bit mathematically. The next thing is I made an equation for the optimal amount of systematic plasticity relative to the unavoidable amount of synaptic fluctuations, so noisy processes, if you like. And it turns out that the optimal systematic plasticity rate is related to the unavoidable fluctuation rate by this constant, which is the upward curvature in the direction of the fluctuation divided by the upward curvature in the direction of the systematic plasticity. And the fact that highly downhill directions are usually curvier basically means that this is generally less than 1. Which kind of proves, well, it's not a proof. You never have actual proofs in neuroscience, but it's an indication of why in generic neural systems, you'd want to have less learning-induced plasticity than synaptic fluctuation if you're trying to preserve a memory, which is somewhat counterintuitive. So in conclusion, for this part of the talk, look at this graph. This graph is the steady state task error. So the steady state level to which I can preserve a memory. And in this simulation, you can see that it's best preserved when the ratio of plasticity rates is less than 1, when there's more synaptic fluctuations than systematic plasticity, so more than the learning rule. The noise is bigger than the learning rule. And the maths is basically to say that this is a general phenomenon that doesn't depend too much on the particular learning rule or the particular circuit structure, and thus gives an explanation for all these confusing experimental results as to why it seems like the intrinsic component of synaptic plasticity seems to be more than the systematic component. It might not be the right answer, but it is an explanation that people can then, criticize or try and improve or whatever. Just to conclude again, to make sure, so we took 3 hypotheses. We said we've got a system where there's some synaptic change that's unrelated to learning and independent of the network. Our second hypothesis was that these changes destroy retained information. And the third was that there's some active compensation mechanism, which you could call learning or relearning. And from those, we got to the conclusion that most synaptic changes should be learning independent. So we were analyzing the steady state regime where I've already learned something, and I'm trying to keep it as best as possible. Obviously, there's another regime where a neural circuit might want to sacrifice retaining previous information in order to better learn new information. I don't care what I've memorized or what's in my thing because it's probably bad. I just want to keep learning and keep absorbing new stuff. So that might be reasonable in an immature animal or a continually learning neural circuit. So the same theory says that in that situation, the rate of systematic plasticity, of learning-induced plasticity, should be higher than the background rate of synaptic fluctuations. So the opposite of what we just said. Now, I showed you this before. This was this cortical, these are these measurements in rat cortex saying that, hey, co-innervated synapses, so ones with the same pre- and postsynaptic neurons, have most of their changes independent of each other, and therefore potentially the synaptic fluctuations outcompete the systematic plasticity. What's interesting is a different lab in a different year did the same thing in hippocampus, and they got a completely different result. It turned out that synaptic changes were very well correlated with these co-innervated synapses. So almost all of the synaptic plasticity was probably attributable to network level mechanisms. It was systematic and coordinated across the network instead of being intrinsic to the synapse. So what maybe we can say, now there's a classical cognitive neuroscience theory of systems reconsolidation, which says that the cortex is kind of the thing that learns in a very slow time scale and is optimized for keeping old information. Whereas the hippocampus is like a scratch pad. It's like a really fast learner, always like learning the next thing and forgetting the last thing. So this is a way of relating that cognitive theory to low-level physiological measurements. Great. What would be really nice though, well, biological data, when you do it in a different lab with different people, comes out differently. So it would be really nice if someone did a systematic comparative analysis across neural circuits and animal ages in the same lab with the same setup, but I'm not going to be the one to do that. Maybe one of you would be, that would be great. The next part of the talk is about learning again. And I'd like to acknowledge Adriana, who was a PhD student I was co-supervising at the time, who was really helpful with the numerics and finding out my mathematical mistakes and that kind of stuff. Yeah, cool. So what did we have before? We had this system, this abstract neural circuit that's trying to retain stored information. And it's got this corrupting effect of these synaptic fluctuations and this restoring effect of the systematic plasticity. Now, this could manifest in two ways if we think about the loss landscape, the error landscape. I could just be dancing around a fixed state. So what does that mean? It means the synapse changes from the fluctuations, the systematic term just undoes it. And it's like, no, come back. Or it could be traversing lots of different equally good states. So the system itself could be changing and changing its behavior while nevertheless maintaining the same level of task performance. Are these experimentally distinguishable? And what would be the scientific implications? I'm going to think a little bit about this now. So when I started my postdoc, I was supposed to try and figure out this phenomenon called representational drift. So we had some collaborators who published this paper, and I went and saw some mice, which was quite interesting. Now, originally, they were looking at trying to figure out what I was talking about earlier, like, can we see the correlates of learning or not learning in a mouse brain, like the changes? And the way they were trying to do this were they were trying to make the mice learn a particular task, which is called the virtual T-maze task. I'm going to explain it. So the mice are in this virtual reality setup, like they've got a VR screen around them. So now, and they're on a polystyrene ball. So they're running, but they're actually stationary. It's just they're running on the ball because the ball is rolling. And then the VR moves with them. So from the perspective of a mouse, I'm kind of running and I'm going through this maze, even though actually I'm head fixed. Head fixation is important because that means you can image the neurons as they're doing all their stuff. Now, the task they had is this T-maze. So you run up the long arm of the T, and you should either go left for a reward or right for a reward. Which should you do? It depends on the pattern of the walls. So if the pattern on the walls is black spots and a white background, you go left. If it's white spots and a black background, you go right. Okay? Differently, they started imaging after the mice had already learned the task. So they gave the mice plenty of time to learn the task. They learn it pretty well. So you can see that the fraction of correct trials they do is pretty close to 1 most of the time. And then they started imaging what's going on. They started imaging an area called posterior parietal cortex. I'm going to use this word necessary, which is a little bit loaded. We don't know how the thing is learning the task. What we can say is that there is this area called PPC, and if you disrupt how it's working, so if you turn off the neurons there, for instance, the mice can no longer do the task. They forget how to do the task. That's not the same if you take off other areas. And the conventional kind of high-level theory is that there's sensory input going through circuits, going to PPC, which is quite a deep area of the brain. And then the decisions are kind of being formulated there, and then that goes to the decision, a motor output. So clearly, if you disrupt PPC, you can no longer make good decisions. Next thing, posterior parietal cortex represents task-related variables. What does that mean? Basically, each horizontal bar in this diagram is the image, is the activity of a neuron over time. So if it's light-colored, that's lots of activity, and if it's dark-colored, that's less activity. And this is averaged over many trials on day one. And what you can see is that there are some neurons over here that fire early in the maze, consistently. There are other neurons that fire really late in the maze, consistently. Now, we don't really know the computational function of these neurons, but the fact it's consistent and it's kind of needed for the task implies there probably is some computational function in terms of making the decision. And at the very least, they're very consistent in the way they fire up. So even if I don't know what the mouse is doing, if I just look at the graph and see, okay, which neurons are firing, I can use statistics to say, oh, the mouse is probably here with a lot of accuracy. So there's enough information just by looking at this neural activity to say where the mouse is. That's one task-related variable, which is how long you are in the maze, but there's loads of other task-related variables, and you can find them all in PPC in a similar way. Well, that's in day one. So this on the top left is the same graph I just showed you. But then if we have the same neurons ordered in the same way and look at their activity on day 10, that pattern is gone. The neurons that used to fire consistently early in the maze or late in the maze are no longer firing consistently at that point. However, if we reorder based on day 10, you can still find that pattern. There are different neurons that are now firing early in the maze and different neurons firing late in the maze. Again, I emphasize we don't know the computational function, but I guess the hypothesis is that there is some stable computational function being done by the PPC since there is stable learning. It's already learned the task, and that bit is necessary for the task. But there's ongoing circuit reconfiguration as that's happening. You can do the same with day 20. So there's always a representation of how far you are along the maze or anything else, to be honest. I only showed this. It's just that representation is changing on a slow time scale, not on a fast time scale within a day. At first sight, this might say that, hey, well, we had this question as, are we dancing around a fixed state? Or are we having systematic changes, ongoing circuit reconfiguration? Maybe that points to the latter being true. But in the end, this abstract task error function is low, whatever it is. But inside, there's actual systematic changes happening to the nervous system. It kind of makes sense because we have to do stable activities even while our brains are changing all the time due to learning other stuff. The problem is it implies redundancy. It implies many different circuit reconfigurations with the same output. And in biology, if you see something that looks expensive from a metabolic point of view, there's a reason for it. Biology does not like to waste energy. And actually, this is on a tangent. This is one of the things that really struck me when I started biology. So one of the first things you learn about when you do neuroscience is place cells. So cells which fire when you're in a particular place. So I probably have a particular cells that fire on in here and other ones that fire on in there consistently. People can figure that out just by imaging like a couple of hundred neurons. Meaning I've got millions of place cells coding for the same place. It's a massive amount of waste potentially. Why would I have millions of neurons potentially doing the same thing? I mean, they might be doing slightly different things, but it seems like there's a load of redundancy. So I want to think about what are the principles? Why would redundancy be a good thing? And I'm going to illustrate that with a really simple numerical experiment. Again, on a very non-biological, easy, artificial neural network with a sigmoidal nonlinearity, a multi-layer perceptron, which hopefully you all know since you're in the AI seminar. So I'm going to take one neural network and I'm going to call it the teacher. The teacher is always right. So we give it inputs, we fix its weights, it does not learn. We give it inputs and we give it outputs. And it gives us outputs. Meanwhile, we have a student. The student has exactly the same architecture as the teacher. So it has the same layers, the same numbers of neurons. And the task of the student is to learn to recreate the behavior of the teacher. So we're going to give them both inputs. The teacher is going to give an output. That output is then used as a label to train the student. So eventually, if we do supervised learning, backpropagation, all that stuff, the student should correctly replicate the teacher's behavior. And the key thing is here is we know the student can learn this perfectly because the student has exactly the same architecture as the teacher. So there is a set of weights for the student which will give you perfect behavior, and that's the teacher's weights. That's just one student, but I'm going to make a family of students. That's the smallest student, but I'm going to make iteratively bigger students, okay? I'm going to keep increasing the student size. And because even the smallest student is exactly the same as the teacher, I know that even the smallest student can perfectly learn the task. So that means all this extra size is redundant in a sense. It's not necessary for learning the task. It's not like you need that extra complexity to recreate the complicated input-output function you're trying to learn. It's redundancy. Then what I did is I gave the students noisy learning rules. So I didn't let them learn with pure backpropagation. I corrupted the backpropagation with noise. And I corrupted them all with the fixed same signal to noise ratio. So like 10% signal, 90% noise or something like that, okay? And with the same signal to noise ratio, bigger ones learn faster and to better steady state error. So somehow the redundancy has made the task easier to learn, even though in a strict sense, it's unnecessary because even the smallest student can learn the task. So what's going on? Oh, actually not what's going on. Maybe this is related to why there's redundant connectivity in the PPC. Maybe the redundancy compensates for some kind of inherently imprecise learning rules because biological learning rules have limited access to information. They have all sorts of physiological constraints. It's unlikely they could do, pure backpropagation if that was even what they wanted to do. So this is a hypothesis. How are we going to make this hypothesis a bit more concrete? Because we just tested this thing in a very simple non-biological neural network. Let's go back to our task error function, okay? Again, I repeat, we do not know what the task error function is for any given neural circuit. We don't know what the loss function is. But we just want to think what aspects of the error function of the loss landscape make it easy to learn and what make it hard to learn. So what makes a landscape nice? Meaning that a noisy learning rule, even if you have a lot of noise, doesn't compromise learning. You can still have fast or high precision learning with a noisy learning rule. On the left is a nice looking landscape. Why is it nice? It's got lots of slope. So even if I go in quite a bad direction that's only weakly correlated with the direction of steepest descent, I'll still get quite a lot of improvement. If I'm skiing down a steep slope, not that I ski, even if I'm going that way, I'm going quite steeply downhill because the whole slope is steep. It also has no curvature. And that means that basically half of directions are good. If I'm going downhill, it's good. That's half of directions. If I'm going uphill, it's bad. So this is a simplification of the maths, but if the slope is large and the curvature is less, it's easy to learn. This on the right is a not nice landscape. It's very shallow. There's not much slope. So if I go in a downhill direction, I don't actually get much improvement. But even worse, if I go too far in that direction, because there's lots of curvature, I'll start going up again. So I have to be very careful about picking the right direction. And a bad signal to noise ratio will really hurt my learning. They're not going in this signal to noise ratio, meaning not going in exactly the right direction for exactly the right amount. And for the nth time, we do not know what the landscape looks like. So maybe this is a problem, right? So what we're going to do is do a trick to get around this. We're not going to ask what the landscape is. We're going to ask how the landscape changes under a transformation. Okay? Otherwise known as the chain rule. So suppose I have an arbitrary neural network with some fixed precision learning rule, okay? There's going to be different weight configurations it could have. So if it had two weights, it could be here or here. Some of them are going to be easy for learning in the sense they have high slope and low curvature. Some of them are going to be hard for learning because they have low slope and high curvature. I don't know which are the easy bits and which are the hard bits. But what I can say is if I take a mapping that preserves the behavior, I can see how that easiness transforms under the mapping. And it turns out when you add redundancy, generically, whether the ratio of slope to curvature gets better. So the second bit, this bit was it was intrinsically hard to learn, but it's a bit easier, though it's still hard. This bit was easy and it's even easier. So we're not looking at what the landscape is, we're asking how it changes under the transformation of adding redundancy. And then we can say something about how learning gets easier without actually knowing what the loss landscape, the error landscape is itself. The mathematical theory and the framework for exploring different architectural transformations is in that paper. So does what I tell you mean that there should be infinite ideal redundancy? We should just get an arbitrarily large neural network to learn everything? Not really, because if you remember, synapses inherently have at least some degree of molecular noise. We don't know how much there is, but things are degrading and changing, and things have to be replenished. If you have a big network, you have a lot of connections. If each connection has an unavoidable little hub of noise, you have more noise as you get bigger. So there's a trade-off. Noisy connections plus more connections equals more noise. So in the end, we get to this trade-off where if we have a really imprecise learning rule, then a bigger network is better because the bigger network has better slope to curvature. If we have a lot of intrinsic synaptic noise, then a smaller network learns best because it has fewer connections for that noise to hurt. We can simulate and make sure that the maths checks out. So NOP does like our mathematical characterization for some really simple circuits of where the optimum lies. Seems to correspond to the experiments, but the numbers are completely unimportant except to verify the maths because these are not biological networks. It's just to say, look, the trade-off exists and we can see that the math seems to check out in simulation. So that's a kind of way in which design trade-offs, we can find otherwise implicit and not necessarily obvious design trade-offs by using optimization theory. So in summary, we had this kind of, well, when I started my postdoc, I was given this data and it was kind of, how do we explain it? There's redundancy and there's ongoing reconfiguration. So the hypothesis is that maybe the redundancy compensates for imprecise learning rules. And the ongoing reconfiguration is because these autonomous synaptic fluctuations might actually want to dominate in a stably learned task like it is. Again, these are hypotheses. Neuroscience is a young science. This is not a proof of these things, but it's something people can go from. At least it's an explanation. I'm not going to talk about my current work, but if I were to criticize what I just talked about, in a way it says everything and nothing. It's not very related to a particular circuit doing a particular task. And so I've been trying to go more into that recently. I'm interested in how heterogeneous architectural motifs represent heterogeneous functional purposes. In the same way a shark looks like a dolphin because they have similar functional purposes, why does a place look different? Does that reflect, can I actually figure out what it wants to do based on that? In particular, something I'm interested in is, suppose I have a limited amount of wiring and complexity I could do. Neurons are expensive. They take up loads of energy. So I don't want to use too many of them usually. If I'm trying to learn a task, I could have an autocracy. I could have one big neural network, one big neural circuit that learns the whole thing. Or I could have a democracy, lots of simpler circuits. And I could allocate extra wiring to making more simple learners or making my one big learner more complicated. Kind of interested in which kinds of learning which kinds of learning problems benefit from either of those. In particular, I'm interested in the Drosophila, which is the fruit fly mushroom body, which is a place where a lot of associative learning happens. So Pavlovian conditioning, that kind of thing. It's quite cool because it has 15 different learners and you can literally see what they are. And they have different personalities. Some are quite optimistic, some are pessimistic, some learn fast, some learn slow. And they all kind of vote in a dynamically weighted way to determine outputs. So as I said, I'm not going to talk about my current research, maybe another time, but some breadcrumbs that I've been working on that might inspire some fun conversations is I think democracies should sometimes be non-Bayesian. I think that there are some biases in democracies that shouldn't adapt to experience, and this is reflected in the fruit fly circuitry. And I'm also interested in principles for simultaneous structural and statistical learning. What does this mean? So we often think of Bayesian inference as a normative principle for how we should learn in uncertain environments. that assumes a task structure. It assumes there's a load of latent states and there's a set of causal relationships relating to them. And those can't have cycles. There's limited number of causal relationships you can have. The problem is an animal doesn't necessarily know that structure. And learning that structure is massively data intensive. So I'm interested in how you can actually, in a data efficient way, not use that structure and actually be kind of therefore be necessarily non-probabilistic, but still react to uncertainty and learn in changing conditions. And that's it. So thank you very much for listening. I'd like to particularly thank Tim O'Leary, who was my supervisor for basically all the work I did and contributed massively to it and was really helpful and a great supervisor. And his lab, who were also very fun. Adriana, as I said, who was a PhD student, really talented, who helped me a lot on the second project. And the collaborators who are responsible. Yeah, thanks. Question time, I guess. Hi.
First of all, thank you for that talk. It was really nice as an engineer trying to do science. That's a nice introduction. More specifically, so you talk about, I really like the result about the network size and imprecise learning rules. So is that network size the number of neurons or number of synapses? Because
synapses.
How does that translate to number of neurons? Because like real biological circuits tend to have lots of neurons but be relatively sparsely connected, so fewer synapses. Is that good? I feel it is good in that framework.
It depends. As I said, if I really look carefully at what I did, I'm thinking about how things get easier or harder in response to changes in the structure, not an initial structure. So if I take an initial structure, which might be sparse, and then I change it by adding redundancy, I can ask, does that make things better? Now, yes, so there's this whole theory of like large sparse networks are good for pattern separation, right? So actually work I've been doing with Adriana recently, and hopefully she'll publish soon, is thinking a bit more about that. Because actually, if you look at granule cells in the cerebellum, which comprise most of the cells in our brain, most of the neurons in our brain, it's not clear whether they are actually sparse. Now, the cerebellum is implicated in motor control. It's not clear that motor control should be good at pattern separation because it's kind of a continuous thing. Pattern separation is like you have small changes in inputs and big changes in outputs. You don't want that in a control system. So we've been trying to use this theory to give a different reason for why redundancy is better that's different from the standard kind of pattern separation view of things. I don't know if that answered your question.
I guess sort of, but I mean is it the number of synapses? Is it having more synapses learning the thing that really matters, or is it the number of neurons that really matters?
Well, I think they both matter, no? I don't think I've given the whole story here, but I'd say that what I'm saying here is that having more synapses than you need and more connections than you need helps. And the way, of course, I was adding connections is by adding neurons, which are connected, right? I was giving a particular architectural transformation that adds both neurons and synapses, and I wasn't speaking as to sparsity. Which is more important? I don't know if that's even a well-posed question, really, but I think that would be very task-dependent.
You have a hand in the Zoom if you want to.
Sure. Okay, I'll go to you afterwards, Greg. Hi.
Hi, Dhruva, it's Nick. That was a great talk. Thanks very much. I liked your introduction section. I'm not sure I could draw such a nice connection between the points in my own career if I was going to do something similar.
Retroactively, right?
Yeah, that's right. I was going to ask you a question after the first part of the talk about if the redundancy in the network would make a difference to that balance between the noise effectively and the network developing, but you answered it comprehensively in the second part of the talk. But what I didn't quite understand in the second part of the talk, you showed, and perhaps you could go back to the slide where you showed the reconfiguration happening over the days in the mouse brain. I think that's it, perfect. I can clearly see the reconfiguration. I wasn't quite sure how that was showing the redundancy. I mean, I can see a bit, I think, but I'm not sure how it's showing a lot of redundancy in that.
For two reasons. One is it's showing that there's different circuits. There's different circuit configurations which have the same amount of learning. So if you look at the loss function, there's lots of places with the low amount of error. So that's a kind of redundancy. The second, which I guess I was a bit more implicit with, is look, they're imaging 100 neurons. And they already see representations of everything. But the actual PPC has, hundreds of thousands of neurons. So the fact statistically you can see all these things in 100 neurons when actually there's 100,000 does also imply some redundancy.
Okay, thanks. Thanks a lot.
Chris, you have a question?
Yeah. So I was curious about the 50% at least in the cortex of neurons that are of the noise that's happening versus the actual synaptic.
Yeah. I'm not sure I should call it noise, but yeah, I agree. Yeah. Okay. Intrinsic autonomous processes. Yeah.
That was kind of my question. It seems like such a massive percent, and then you're linking it to degrading performance. But I guess are there theories around how it's helping performance, or does that change anything? Since it's such a large percentage of synaptic change, does it suggest that it's probably beneficial to the circuit in some capacity?
I think that there are certainly reasons it could be beneficial. One is when you're learning, you might want to have a kind of exploration where things change to get to different circuit configurations. I guess I'm not speaking to that. When you're doing neuroscience, you kind of have to focus on one particular functional purpose. The way I was conceptualizing this is something that is unavoidable to an extent. Now, neurons can reduce this. Some neurons are very stable. Some connections are very stable. So it's physiologically possible. But a nervous system always reduces noise or these kinds of things to the extent it needs to, because you need to invest more effort so the more you reduce something, right? Given there is this amount of unavoidable noise, how much compensatory systematic plasticity should there be? All I'm saying is in that situation, there's less. And that would be a reason for why in a generic circuit that stably learns something, you should actually imagine that these synaptic fluctuations are contributing the bulk of synaptic plasticity, whatever that level is, whether it's high or low. And maybe in some circuits, it's fine for it to be high. In some circuits, it's fine it needs to be low. I guess if you were going to the lost landscape kind of interpretation, if something hasn't been learned very well, so there's still loads of room to improve more, you're much more forgiving of changes in the synaptic weights than if you've learned very highly to the extent the circuit is capable of. Does that answer the question?
Yeah, it makes
sense. Hi.
To get back to the spontaneous fluctuations versus systematic plasticity, is there any sense where that could be interpreted as a kind of robustness thing? So noise in some dynamical situations tends to have a kind of stabilizing influence. So is there any sense in which just having that...
You mean like a Turing pattern kind of thing?
Yeah, so is there any sense in which having a lot of spontaneous fluctuation kind of prevents systematic effects from kind of becoming unstable and sort of running away with themselves? So you tend to end up with scenarios where there's...
With a kind of epileptic-like scenario, you mean?
Yeah, just I think, it's a kind of common theme across control.
Potentially, but I think when we're thinking about that in control,
yeah,
it's usually fast positive feedback loops that have those problems, right? So that's in activity space, in neural activity space, if you like, right? I feel like synapses are slow enough that, that's less of a problem potentially, but this is very woolly. I'm not giving you a mathematical proof of this. This is just a gut feeling.
Okay. It's just a thought if there was any sort of parallel between the role of noise as a kind of stabilizing mechanism and what you see in terms of... I don't think we really know about stability in plasticity rules. I mean, there's this paradox at the heart of a lot of theoretical understanding of synaptic plasticity, because we have these Hebbian plasticity or approximations thereof are often thought of as the substrate of plasticity. But I think if you actually ask an experimenter who's doing it, they'll tell you like maybe 1 in 5 neurons sometimes is actually displaying that. The problem is Hebbian plasticity is inherently unstable. And if you look at the compensatory mechanisms, synaptic scaling and stuff that people have found that kind of reduce that instability by normalizing things, their timescale is too slow. Like mathematically, they aren't enough to deal with it. I think it's probably because Hebbian plasticity and that kind of thing is not the whole story about in terms of synaptic plasticity and learning rules. But yeah, I mean, I guess that's obvious in the sense, but like, yeah. But yeah, maybe, sure. I don't know. But my gut feeling is synapses changes are quite slow. These things are happening over, hours. So it's not so worrying for fast positive feedback loops, but I don't know. Hi.
Yeah, so I was wondering, same bit of the talk. Yeah, so you did this with the perceptron in the background, right? As your test system. Have you got a good feel for how general these results are? How much it depended on where you were testing them in, or is that
well, that's the optimization theory past the task, because I completely agree that a perceptron is not good enough really to show the thing. So the optimization theory is kind of saying that generally, if you add a new neuron, you're going to make the slope better than the steepness. I can give you a bit of mathematical intuition if you like, which is like, so if you have the gradient, which is the slope, and you have the second derivative matrix, the Hessian, which is the curvature. How much the curvature annoys you is related to how much your direction projects onto the Hessian, which is roughly related to the average eigenvalue of the Hessian, or at least it doesn't scale too fast away from that. If I add more things, the average eigenvalue doesn't necessarily get bigger, whereas the slope is related to the L2 norm of the gradient, so that kind of gets bigger with more. Very rough.
Yeah. So would you say that this, these sound like properties that anything you can describe as some sort of dynamic system, or?
I think the difficulty with, I guess I just haven't really explored it, is I looked at a particular transformation of adding a neuron in a layer. Yeah. I think you could do the same modality for all sorts of different transformations. I haven't done it. Yeah, but basically the chain rule. Take your network, add your redundancy, or change your network property in the way you like while preserving behavior, and see how the less landscape things change under that transformation. Hi.
So I really like this idea of looking at the trade-off between having multiple, let's say, simple learners compared to one much more complex learner. Yeah. And I presume there's some sort of spectrum in between versions as well.
Definitely.
What would be the way of actually, like, how would you quantify this sort of your position along that spectrum? And in particular, with the Drosophila, all of these 15 or however many compartments there are, they are also highly interconnected through other sort of neurons outside of the mushroom body. So
there's loads of recurrent connections.
A way of quantifying this in a nice artificial system, which is perhaps easier to grasp, but also do you have an idea of how you might go about doing that with the actual neuroscientific data?
Yeah. So I'm trying to predict actually some of the recurrent interactions in the mushroom body, in particular learning paradigms, like we talked about second order conditioning, for instance, right? Basically, when I'm doing that stuff with the mushroom body, I'm already assuming it's on one side of the code where it wants democracy versus an autocracy. Because I think an autocracy is good when your principal problem is the precision of your learning rule. Whereas a democracy is good when the principal problem is lack of information. And I feel like the Drosophila is kind of more in the lack of information thing. It's only learning simple things, but it just doesn't have enough information to know when the reversal happens and when it should change, right? I haven't built up that framework properly. That's something I'd like to do. But I'm already assuming that, like, the Drosophila has this many compartments. They're learning with different biases. And then the question is, what principle, if I can say what principles imply that they should be learning with these biases, I can say something about how they should talk to each other to do an overall behavioral output. So why should they talk to each other? There's a couple of reasons. One is that sometimes bad learners need to be taught to be good learners again, but keeping their biases. Another is that I guess I'm talking to you more than the audience now, because but we often use very simple conditioning paradigms. Pavlovian, stimulus goes to reward, right? Whereas a real animal has a very complicated spatiotemporal set of stimuli, right? So it's got this constant problem of, I'm seeing something that's not quite the same as before. Is this a new context? Or is the similarity with previous contexts actually indicative of the fact that I should behave similarly to it? And I have been going down this route and saying, actually, just by thinking of these learners as a way of holding those simultaneous hypotheses concurrently, so a familiarity learner, if you like, and a novelty learner, you can predict a lot of interactions between the mushroom body, some of which are validated, and get reasons for why they should talk to each other. Sorry, it's a bit woolly because I guess I didn't have any slides on this, but we can talk about it in more detail later if you like.
another online one.
Sure.
Simon.
Hi.
Simon, do you want to just speak?
Sure, can you hear me?
I can hear you.
Great. Yeah, thanks for the talk. I really liked it, and I'd be interested in learning more about the optimal non-Bayesian updates maybe at some point.
Yeah, I'd be happy to just chat whenever. Yeah.
But I have a question like Lionel. I have a question about whether there might actually be a functional feature to neurons doing their own thing, which is that there are examples in bounded resource optimal control where non-deterministic strategies are just better than any deterministic strategy. Could that be another possibility? So this tends to be like a compensation for limited memory, although I suspect it's also, it'll also be the case in sometimes when you have bounded processing speed or bottlenecks or something like that. Could that be an alternative possible explanation for?
Well, certainly, there's many reasons that would probably happen. I don't think it would be truly exclusive, right? I think I'm just concentrating on one, right? It certainly doesn't, like what I've said, does not discredit other reasons for the noise to be there. I mean, if there's another reason for it to happen, then great. I don't really know enough about that particular branch of optimal control to comment intelligently on whether that's good or bad, but yeah.
All right, thank you.
There's something in the chat as well. Thank you. There's someone in the chat. Oh, it's the same. Okay. Great. Do I get to have my nap? Oh. Yep. Thank you very much for coming, everyone.